Thanks everybody for coming to the webinar today about HLVd. We just thought we'd put this out there. HLV has been a problem in the industry since about 2020. So we've been dealing with it in Canada for about 6 years. Still lots of problems. We still detect plenty of it. So this is just a quick session. Again, a lot of people already know a lot about HLVd who had to deal with it. We shared lots of information. Um, Keri Wang, Dr. Keri Wang here. He's going to be giving the presentation today. He's our leading expert. Keri, for you don't know him, Keri's been with us about 17 years. He developed all our disease diagnostics in A&L. So he personally has made a lot of the detection methods. He's our leading expert in plant pathology. He's a geneticist. Um, you guys, can reach out to him anytime. He's a great resource. You can send pictures, and Keri and a nutrient agronomist will look at your images and help you decide how to test. So, that being said, I think we'll just let Keri take it away and do his presentation. Thanks, Keri. Go ahead.
Okay. Hello, everyone. So first, let me share the screen. Okay. Good morning, everyone. Welcome to this presentation. So, yeah, I'm Keri. So I'm working on microbiology and plant disease diagnostics. So today I would like to talk about one of the most disastrous diseases, Hop Latent Viroid in cannabis. So first of all, what is HLVd? So HLVd is a single-stranded RNA. So the genome has only about 256 nucleotides. This does not protein was hop plant. So this disease is very common worldwide, wherever hops are grown. So in cannabis, HLVd was reported in California in 2019. In Canada, A&L was the first laboratory reported HLVd in cannabis in 2020. So HLVd could cause severe symptoms, but sometimes the infected cannabis plants may not show any symptoms. Anyway, regardless of the symptoms, HLVd could cause about 30 to 50% yield loss and 50 to 70% loss in THC content. So what are the differences between virus and viroid? So all viruses have the DNA or RNA genome and code protein here to protect this genome. So viruses can infect all the living organisms, including our humans, animals, plants, insects, birds, even fungi and bacteria. However, viroid has only a small piece of RNA genome. So it does not have any code protein, does not produce any proteins. HLVd can infect the plants only and cannot infect any other organisms. Since we identified HLVd in cannabis in 2020, we tested about 5,000 samples every year. In 2020, we received over 2,000 samples from British Columbia, where it had HLVd outbreak. So about 92% of the samples were HLVd positive. Then in 2021, we received over 8,000 samples, but only about 16% samples were HLVd positive. So from 2022 to last year, the infection rate was reduced from about 15% to about 8% last year. So this could be due to the application of good control measurements. So what are the symptoms? HLVd can induce many different symptoms on mother plants. For example, these curling leaves, okay, this one and this one, and also the striking on the young leaves. And some infected plants show stunt growth with shortened internodes and smaller leaves. Just like this is infected one, and this is a healthy one. Some genotypes also showed sideways or lateral growth, like this and this one. And the infected plants may also show inward curling. You can see like this one. Okay, as I said before, the infected plants may not have any symptoms like this one. However, when we took 4 samples from different parts of that infected plants, like this 1, 2, 3, and 4, all of these samples showed HLVd positive by RT-PCR. Now let's see the symptoms on inflorescences. So this left are healthy, okay, this red are infected, HLVd infected. So you can see the infected inflorescence are much smaller than the healthy one. The thread inflorescence also, smaller compared to the healthy one. So in addition, the inflorescence may also develop the colored leaves, such as, see this one, yellow, or the purple leaves. HLVd also affect the trichomes. So this left also healthy, the red are HLVd infected. So you can see the infected inflorescence developed much smaller trichomes. Under microscope here, and you can see clearly the healthy inflorescence had fully developed trichome heads with the long stalks. The infected one had short trichome stalks with smaller heads. So next question you may ask, where HLVd is in a plant? So before answering this question, we did a study to show where virus is in a plant. So to find out where the virus is in a plant, we labeled a virus using a green fluorescent protein so that the virus in plant can be visualized by the naked eyes under a UV light. So here we inoculated the tobacco leaf with the labeled virus. So we can see the virus infection on the inoculated leaves as green under UV light, but not on other leaves. So however, the virus can quickly spread into roots. So we can see some roots were green already here. So these green roots means virus infection. A few days later, the virus spread to the whole leaf and some other leaf here, but not all other leaves. And some other leaves, still do not have any virus infection. However, most roots were infected with viruses. So you can see here, most roots were green already. Then virus keep spreading to other branches. For example, here, we inoculated here and also spread to this branch. But not all other leaves. So these leaves still did not have virus infection. However, check the roots. All roots were infected with viruses. So viruses move to roots faster than to other leaves. So this type of spreading is the same for HLVd. Different branches of the same cannabis plant could have different HLVd infection. So this plant showed that top branches were HLVd negative, and the lower branches were HLVd positive. So you can see the different inflorescence size. So the healthy inflorescence are larger, and these infected ones are smaller. So, and also the healthy trichome also are larger and have the higher HTC content, about 28% compared to the infected one, only about 16%. Because HLVd distribution in cannabis plant is not even, the detection success will be different when taking samples from different parts of plant tissues, especially from the newly infected plants or young plants. Roots, where the roots always gives the most consistent HLVd detection. However, the above ground tissues could have about 36% to 75% success of for the newly infected cannabis plants. For sure, when virus has well established in a plant, all tissues will be HLVd positive, regardless above ground or underground. So how HLVd is transmitted? HLVd is primarily transmitted mechanically through the roots contaminated, tools during the pruning and handling. Vegetative propagation is also a major spreading means. So the transmission rate through the infected mother plants can be as much as 100%. Since HLVd is a seed-borne pathogen, so the infected seeds are the main source for the new cannabis facilities. HLVd can also be transmitted through root-to-root contact and recirculating hydroponic systems. Good thing is that no insect vector has been reported to transmit HLVd. Okay, as you know, HLVd can be detected by PCR only. Currently, there are two types of detection kits available on market and used by many laboratories for HLVd testing. So the first one is the Green qPCR. So while this is widely used, it has some notable limitations. First, the sensitivity is relatively low. It cannot detect the targets with low copy numbers. In addition, the major drawback is low specificity. Green dye combined all double-stranded DNA, including the non-specific PCR product and even the prime demo. So the green qPCR could lead to false positive. The second one is Tegman qPCR. Tegman qPCR uses a Tegman probe to detect the PCR products. The probe is labeled with two dyes. However, when these dyes can be degraded over time of storage and repeated freeze-thaw cycles and lead exposure. So once these dyes are degraded, it will lead to reduce or even lose the detected signal. So therefore, the Tegman qPCR could lead to false negative. we developed our own method for HLVd testing. So we use reverse transcription PCR and capillary electrophoresis technology. So first, here the PCR product can be visualized, to identify the specific PCR product. So for example, in this positive control, we can see this is the band for HLVd, and this one is the internal control. So this PCR product also can be quantified. So this signal here is from the HLVd specific band. This one is the internal control, the signal for the internal control. So in the negative control, there is only the internal control band, no HLVd band. So the internal control is to make sure the RT-PCR works for each individual samples. We also include a positive control and a negative control for each set of RT-PCR. The negative control is to make sure no contaminations. Positive control makes sure no false negative, and HLVd detection works. So our method is very accurate. To test the sensitivity of our method, we made a series of 10 times dilution of the total RNA extracted from the infected cannabis leaf sample. So we can see HLVd can be detected after here the 10 times dilution, 100 times dilution, 1,000 times dilution, and even after 10,000 times dilutions. So our method is very sensitive for HLVd detection. To give you a general idea about HLVd levels in samples, we report the results semi-quantitatively based on the signal strength of RT-PCR. For example, if no HLVd detected, so we report it negative, and here is we report very low, or here is low and moderate, and is high. So when we detect HLVd very high, we just report it as high. So then, how to take samples for testing? Since HLVd is transmitted mechanically, so it's very, very important to wear gloves when taking samples, and change gloves or spray with 10% bleach between plants to avoid cross-contamination. If you take leaf samples and test an individual plant, please take 5 to 10 leaves from different branches to cover as many infected leaves as possible and to make the detection successful. If you want to do the screen of the many plants, then you can take about 2 leaves per plant and composite 10 leaves as one sample. Since roots are more sensitive for HLVd testing, you can take about 3 to 5 root segments from the crown area, and then put the samples in Ziploc bags, seal it and label well. Ship the samples by the express mail. Last, how to manage HLVd. So it is very critical to start HLVd-free mother stock clones. Start clean and keep it clean. So to start the clean, the mother plants need to be tested every 4 to 6 weeks and before cutting. Please take roots for mother plant test. To keep clean, the productive clones can be pulled together for testing to cover as many plants as possible and to save the cost. You can take leaves for screening of the productive clones. Since HLVd is pretty stable in the environment, the services and the tools need to be monitored. So you can swab the environment and send it to us for the testing. All incoming cannabis plants into the facility need to be tested and keep quarantined for 30 days. Once detected HLVd positive, destroy all the infected plants and isolate the infected rooms. So last, keep good sanitation using 10% bleach or 2% worker. Okay, thank you. I'd like to take all the questions.
Thanks, Keri. That was great. Yeah, if you guys have any questions and you want to put them in the chat, or you're welcome to email them to us later, or reach out. That's totally fine. Yeah, we really wanted Keri to highlight kind of how detection moves with the plant. So his example of the virus fluorescence is a great example. Some of that research was done on HLVd in a way to figure out that HLVd was indeed the root. So, we want to highlight the root sampling when possible, especially on the mothers. Good to have a great mother program to quarantine your mothers and test them. Plants, can finish out their cycle if they're infected with HLVd, and you may or may not get reduction in yield and THC content, but after that, you would want to, clean that room and make sure you don't infect your mother. So that's super important. We see that all the time. Quarantine the plants again before they come to the facility. Time and time again, I see people who don't do this, or they're told that the plants were tested and they weren't tested properly, or the plants they got were infected and the plants tested were not, because it's not everywhere. So protect yourself and quarantine those new genetics coming in. Yeah, I think that's about it. If there's no questions, reach out. Keri's always available. We're always available here. So appreciate you guys coming. We plan to have some more disease related seminars in the future. And yeah, thanks everybody. Have a good day.